Hello, everyone in Japan, and good morning to everyone in Switzerland. My name is Kyoko Marumo Suzuki. I'm the head of the Science and Technology Office Tokyo at the Embassy of Switzerland in Japan. I would like to thank everyone for joining us today, especially those in Japan who had probably a beautiful autumn day, hopefully. At least in Tokyo, it was very nice and sunny on this public holiday. The audience is very diverse today, from young students to experienced adults, as well as researchers and media experts. Our mission at the Science and Technology Office Tokyo at the Embassy of Switzerland is to connect Switzerland and Japan in the fields of education, research, and innovation. So I would like to thank everyone joining, the audience, the speakers, and organizers on this special international session at Science Agora 2021, which fulfills part of our mission. We have an exciting and full program today. So without further ado, I would like to welcome Dr. Christian Sim, Head International Relations Office at the University of Zurich. Dr. Sim.
Thank you very much, Kyoko, and a very warm welcome from everyone to everyone here in Zurich, where it's not that sunny. It's actually pretty gray, but we're quite excited to be in connection with you. I just wanted to welcome you all, give you just a few words about Switzerland and the University of Zurich. You know, Switzerland is a small country in the middle of Europe. In many ways, actually, we have lots of similarities with Japan, because we both very precise, very clean, very detail-oriented research and manufacturing. I think in that sense, we have a common mindset that creates lots of great collaborations between our two countries. Switzerland is consistently ranked as number one in the most innovative countries in the world for the last 10 years at least. And inside of Switzerland, a country of about 8 million, the University of Zurich is the largest and the most diverse, comprehensive university. We have 7 faculties, 150 institutes, like 28,000 students, 720 and more professors, and nearly 7,000 research staff and support staff. So it's a very big institution that studies everything from theology to high-end medicine, from economics all the way to physics. And it's actually, physics is and has been in the past also a very important subject at the University of Zurich, because we actually have 12 Nobel Prize winners, and one of them, and probably the most famous one, is Albert Einstein. But we have a few others like Wilhelm Röntgen, who discovered the X-rays. So just to tell you that it is a really great, not only research university, but of course also a university to learn a new profession. Very strong fields at the university are, of course, it's always difficult to make a list, because then you don't mention everyone, but it goes really from linguistics to behavioral economics, from financial market research to genetics, structural biology, medicine. We have also the biggest medical school in Switzerland. And scientifically, I think we can be quite proud about the results that our researchers produce in their research papers, since we are in the highly cited publications really very much on par with our neighbor ETH Zurich and the University of Oxford, for instance. And we're also a very international university. If you go into at the level of the non-professional academic staff and the professors themselves, we have actually about 54% of people that are coming from abroad. So a very welcoming place, a very nice, very interesting place, Zurich. And we would welcome you in Zurich as well. If you're ever interested, you can always contact us. And with that, I think you want to listen to the scientists, to the specialists of these very fascinating fields. So I thank you very much for listening. I wish you a very interesting Science Agora and a good day. Thank you.
Thank you, Christian, for the very concise overview of University of Zurich, which we are very proud to present today, researchers from University of Zurich. The title of our session today, excuse me, is From Earth to Space to Earth, from regenerative medicine to biodiversity. So we shall start from Earth, and I would like to welcome Dr. Christian Tackenberg first. He is the scientific head of stem cell research at the Institute for Regenerative Medicine, IREM, University of Zurich. He is also group leader at the Neuroscience Center Zurich and a lecturer for the Faculty of Science. And his research aims to help us fight Alzheimer's disease and recover from brain diseases such as brain strokes. So very important, to say the least. Dr. Tackenberg, thank you for joining us today, and we look forward to your presentation.
Thank you very much, Kyoko. So good morning from Switzerland. I hope you can see my screen.
Yes, we can. Thank you.
Perfect. So thanks again, Kyoko, and thanks also to the organizers of Science Agora for the invitation and for giving us the opportunity to present our research here from Zurich for you. Before I start with going into detail about our research, I would like to quickly give you an overview about the institute where Dr. Generali, who is giving the second presentation today, and I are working, which is the so-called Institute for Regenerative Medicine, the IREM. The IREM was founded in 2016, so it's a relatively new institute, just 5 years old. And the mission of our institute is to bring together researchers from various areas to synergistically aim to understand various diseases. And based on this understanding, we want to develop next generation therapies. So here on the right part of the slide, you see the various areas that we cover at our institute, which range from cardiac regeneration, tissue engineering, to gene therapy, to mechanisms and therapy of various brain diseases. Kyoko already mentioned stroke and Alzheimer's disease, imaging biomarkers, brain and spinal cord regeneration, and the regulation of gene expression in healthy and diseased tissue. We also have 2 clinical centers associated. One is the Center for Therapy Development, GMP, and one is the Center for Prevention and Dementia Therapy. And recently, we were also able to establish a core facility for induced pluripotent stem cells, iPSCs, that is headed by Dr. Generali, who will give the second presentation, as already mentioned. Okay, but coming back to my field of research. So I'm a neuroscientist. This means that I'm interested in how the brain functions and how to cure dysfunctions of the brain. So I think the brain is really a wonder. So it controls all our body functions. It is important for our personality. And it is regarded as one of the, if not the most complex structure that we know in the whole universe. The brain consists of 86 million neurons, so these are the nerve cells, and around the same number of non-neuronal cells. These neurons are connected through around 100 trillion synapses. And also the energy demand of the brain is really amazing. So it weighs only around 1.5 kilos. So this means it makes up between 1 to 2% of our body weight, but it consumes between 20 and 25% of the energy of the whole body. And this energy supply is secured through blood vessels that transport all the nutrients like glucose and also the oxygen into the brain to ensure proper brain function. The problem is now if this supply does not function anymore. For example, if a blood vessel is clogged through a thrombus, and this causes a so-called ischemic stroke. So this means there is an area where there is not as much supply especially with oxygen as it is supposed to be. And this causes the cells to die in this area. So currently, we only have two treatment options. One is the thrombolysis and one is the thrombectomy. So this means that we simply remove this thrombus to make the blood vessel permeable again. However, this is only an acute therapy, which has to be carried out within the first 4 to 6 hours. And what you have to keep in mind is this: within every minute where a stroke is not treated, around 2 million of these nerve cells die, and around 14 billion of synaptic connections are lost. So this means that even if we treat these patients within a few hours, there is a lot of neuronal degeneration. Thus, there is a very high demand for a potential regenerative therapy after stroke, which is currently not available. And we think that cell therapy, so this means the addition of newly fresh functional cells to the brain, would induce neuronal recovery after a stroke or after other brain diseases. But the question is, what is the right cell type to add to the brain? And therefore, it is important to understand what cell types do we have in the brain. I already mentioned the neurons, or the nerve cells, where we have around 86 billion cells in the brain. However, we also have other important cell types, which are the so-called glia cells. Glia cells are astrocytes, microglia, or oligodendrocytes. And all cell types are very important for the function of the brain. Astrocytes, for example, are important for the nutrient support and also for the synaptic function of the nerve cells. Microglia function as kind of immune system of the brain, and oligodendrocytes, you see here, they enwrap the processes of the nerve cells. They are essential for a fast information transport. But not only these 4 cell types we find in the brain, we also find so-called neural stem cells that are abbreviated with either NSCs or NPCs. NPC stands for neural progenitor cells. So we have 2 areas in the brain where we find this cell type, and these cells can give rise to new and fresh neurons, astrocytes, and oligodendrocytes. However, the number of these NPCs or NSCs that we have in the brain is so low that the brain cannot regenerate itself. So we have to help the brain to regenerate. So our idea is to provide the brain with new neural stem cells. But also then is the question, where do we get these nerve cells from? Because we cannot simply take them from a healthy individual. And the solution is from induced pluripotent stem cells, short iPSCs. This is a technique that has been discovered by Professor Shinya Yamanaka from Kyoto University in 2006-2007. Professor Yamanaka found that when he expressed certain factors that are now also named after him, so-called Yamanaka factors, in somatic cells, so he first tested this with skin cells, with fibroblasts, that he could reprogram these cells into a pluripotent cell that he then called induced pluripotent stem cell. And every pluripotent cell can give rise to basically every cell type. So it can differentiate into a nerve cell, into an astrocyte, into a cardiac cell, so into a heart cell, a skin cell, a liver cell, whatever. So besides the use of modeling and understanding diseases, these iPS cells have a huge potential for cell therapy because we can basically generate every cell type that we want to use for regeneration. So at our institute, we have a quite big project running, an interdisciplinary project running between neuroscientists and the cardiologist. This project also involves a very close collaboration with our colleagues from the CIRA, the Center for iPS Research and Application. This is the institute led by Professor Yamanaka at Kyoto University. So what we aim to do is the following. So for both projects, we start from the same iPSCs that were generated by Dr. Generali. These iPSCs are either differentiated into cardiomyocytes with the aim to regenerate the heart, for example, after a myocardial infarction, or, and this is more my part of the whole story, we differentiate the iPS cells to neuroprogenitors that are then transplanted into the brain to regenerate the brain after a stroke. Of course, we cannot directly start with a human patient. So what we do is we use a mouse model for ischemic stroke, which is called the photothrombotic stroke mouse model. It's relatively easy to explain. So the mice are injected with a substance that is called rose bengal. The substance travels through the vessels in the brain, and once it is illuminated with a certain wavelength of light, then it begins to clog the blood vessels. So this means that we can really adjust the area where we induce the stroke. In our case, we induce the stroke in the right brain hemisphere, in an area that belongs to the sensory motor cortex. So what you can see here is a perfusion image. So this means if the brain is shown here in red, this is from the upper side, then there is a very good blood perfusion. This is before the stroke. And after we've induced the stroke, you see here the right side of the brain that there is a strong reduction of blood perfusion. So similar to what is found in human patients. We also see massive tissue degeneration. Here you see the loss of neurons and alterations of the blood vessel system. And we see a leakage of the blood-brain barrier. So I mentioned we induce the stroke in the motor cortex of the mice. So this means that the mice have locomotor, so movement deficits, so also typical to what is found in human patients that suffered from a stroke. But it is not that easy to determine, especially subtle locomotor deficits in mice. And we do it with the following technique that we've recently established. So what we do is we film a mouse in high resolution from 3 different angles, so from below, from the right, and from the left. Then we enter this movie into a software that is based on deep learning, so the software can learn itself, and it is able to track various body parts of the mice. So for example, the tip of the tail, the nose, and all the other areas that you can see here. So the iliac crest, the hip, the back ankle, the back toe, and so on. And with that, I hope you can see the mouse running now. With that, we are able to see, for example, if the mouse after a stroke makes smaller steps or is not able to lift the shoulder or the toe as high as it was before. So I just show you 2 examples. So here we analyzed the back ankle and how high the mouse is able to lift the back ankle. And you see that this is the baseline, so before stroke, that after the stroke, the mouse is not able to lift the back ankle as high as before. And similar findings we have with the shoulder, for example, that the shoulder is not able to lift as high as before the stroke. And we think that this is really a great tool to look for functional recovery after we've transplanted the cells. So in the last 2 slides, I want to show you how we transplant the cells. So what we do is we use our neural progenitor cells that we obtained from induced pluripotent cells. These cells are modified, genetically modified in a way that they express a certain protein that is called luciferase. So then we transplant the cells into the area of the stroke, around 100 to 150,000 cells in the brain. And this luciferase protein is able to emit light if it is in contact with a certain substance. And this means when we place the mouse in a bioluminescence imager, we can measure the light that is emitted from our cells. And this you can see here. So these are around 120,000 cells transplanted directly or imaged directly after the transplantation. And we are able to follow the transplant over the time course here of 35 days. So this means this is a non-invasive imaging tool where we can really follow up the localization of the cells that we transplant and also the survival of the graft. So we analyzed the fate of the cell. So this means what happens to the transplanted cells, in this case, 5 weeks after the transplantation. So very important to mention is that we did not find any signs of tumor formation, and there are no residual pluripotent cells. This is always important because one of the limitations of iPS-based therapy is its potential tumor formation. but we do not see any kind of tumor formation. We see that some cells stay as neuroprogenitor cells, but the vast majority of cells differentiate into mainly nerve cells, so neurons that you see here in red, some also to astrocytes. So this is now an image from a healthy mouse. The next step, what we are currently doing in the lab, is we transplant these cells into stroked mice and see whether these cells can really functionally integrate into the brain network and cause a functional regeneration of these movement deficits. So maybe at the next Agora meeting, I can present to you whether these cells are able to regenerate the brain or not. So I want to close my presentation with thanking all my colleagues from the Institute for Regenerative Medicine, especially Ruslan Rust and Melanie Generali, for doing a lot of the work that I showed you. I want to thank all my colleagues from the CIRA. Maybe some of them are also listening now to my presentation. And Dr. Generali, in her next presentation, will give you some more insights into this collaboration. So with this, I want to thank you for your presentation and say, go seicho, arigatou gozaimashita. Thank you very much.
Thank you very much, Christian. It's very impressive what you do, and we definitely have to do a follow-up meeting now, a presentation now with your next achievements. Thank you very much. As mentioned in the program, we have a short Q&A session now. If people would like to ask questions, there is a Q&A tab, and also we can also accept questions from the chat function. And I do see one question already, which is very nice.
Perfect.
How transferable is the knowledge gained from animal studies?
That is always the big question. I mean, we have this problem for two different areas. So on the one hand, for the area of therapy, I mean, we have to validate therapy in animal models, of course. And for this, we probably mostly use the mouse. But also for understanding diseases, it is always problematic because all our knowledge that we enter when we make a new therapeutic approach is based on knowledge that we usually gain from animal models or from cells from animals. And also, therefore, these iPSCs are very, very valid tool to better understand how a disease functions in a human cell. Because it is known from several studies that when you take, for example, neuronal cells or astrocytes from a mouse, you culture them, and you compare this to human cells in culture, these cells can behave really, really different. So I think we will always need mouse models for both, for understanding diseases, but also to develop a therapy, because what we cannot have from the iPS cells is the whole systemic effect, how an agent behaves in the bloodstream, how long it is in the system, when it is cleared out from the system. But I think the iPS cells are a very good tool to complement mouse models. But in general, it's always a problem to transfer the findings from an animal model to the human patient. Yeah.
Nice. Very interesting. Thank you very much. I would like to remind the audience that we can accept questions in the Q&A and also chat functions. We have another question, actually, or comment question from Mr. Karagianis. I hope I pronounced it.
Hello, Peter.
Christian, it is wonderful to see you, if only virtually. Do you have any plans for controlling the ratio of neurons, astrocytes, and oligodendrocytes after transplanting the MPCs? Thank you.
This is really a very important point. So we currently see that when we transplant the cells in healthy mice, that we have mostly neurons. But it is known, especially from in vitro studies, that the level of oxygen that you have in the system also determines how many or what is the ratio between glial cells and neurons that you get. So we are currently transplanting the NPCs in the stroke mice where you have a lack of oxygen. So it may well be that the ratio of neurons to astrocytes is different. You can control this maybe by manipulating the cells before they are transplanted, by either culturing them in higher oxygen and lower oxygen, so to prime them a bit more. But this is a very, very crucial point that we're currently looking into. Yeah. Thanks for your question, Peter.
Thank you. Actually, there was a little bit more to the question, and also we have a second question. However, since we are tight with time, I would like to save these questions for the dialogue session that, if you don't mind, thank you. For the dialogue sessions, please, Mr. Yamauchi and Peter, if you cannot stay, please let us know. But hopefully you can stay for the dialogue session so that we can start with your questions. So with that, thank you very much, Christian. And we look forward to the discussion session with you after the presentations. Following Christian's presentation, I would like to welcome Dr. Melanie Generali, head of iPSC core facility, also at IREM. She works on a collaborative project with Kyoto University as well. And her presentation title, Mending Broken Hearts, already highlights how the research can help us. Dr. Generali?
Thank you very much, Kyoko. So it's a pleasure for me to present you our research to you today. And thank you for the organizing committee inviting us. So, and for me, it's of particular interest to present today because we have a close connection between our Institute for Regenerative Medicine to the Center of iPSC Research and Application, as Dr. Tackenberg already mentioned. We started in 2017 with an initial collaboration kickoff in order to figure out where are the overlaps, how we can work together. And in 2018, some of our researchers traveled to Japan and we just discussed about possible collaborative projects. And I must say in 2019, we had a really active exchange. It was a really nice year. Visitors as Peter and Saito Sensei visited us in Switzerland. And also a colleague of mine, Deborah Kehl, and me, we did a research internship at the CIRA in this year. Luckily, we were also invited to observe the production of the iPSC-derived cells for clinical application, which was an exciting event. And at the end of 2019, several researchers from Japan visited us in Zurich for a workshop, and we had a really fruitful discussion how we can collaborate together, and also to see that there are quite overlaps between the research in Japan and also Switzerland. And last year, this was a really important step between our universities in Zurich and Kyoto. So there was an official collaboration agreement for a strategic partnership. And despite the current situation, because COVID is influencing all of us, I'm really happy that there are virtual platforms such as Agora 2021, where we can present our research. So, and I would like to give you a broad overview about our research in the field of cardiovascular research. So in 1989, the Life magazine published this picture of possible replacements of injured tissue and organs, so-called the Robocop, because these replacements were mainly ceramics, plastics, or also metals. This concept was quite accepted by the scientific community. And just 10 years later, in 1999, so they published a similar picture, but this was a bionic human, meaning that there was a shift of this accepted concept changing from ceramics and metals to biological substitutes in order to avoid immunogenicity reaction and also to have a kind of grow in the human body. There are several application possibilities. For example, tissue engineered skin are already in clinical use, but also artificial bone pieces are under production. Vascular grafts, for example, are also already in use for dialysis patients. And also we are able to produce nerve grafts in a petri dish. I think a quite hot topic is also the meat analogs. So we will see if this is really something for the future for us, what we will eat in the future. Furthermore, the trachea and the urinary bladder, they are still under preclinical evaluation. And I would like to show you how we are working and how we develop the heart valves. Currently, heart valve replacements are more or less the mechanical ones. They are still in use, or the bioprothesis, meaning that this relies on, or they are derived from pigs or cows. But there are several limitations, like the calcification, the immunogenicity, also the risk for infection, thromboembolism, and the main advantage, this is not a real solution for younger patients, so there's a lack of growth. Therefore, tissue engineering represents a possible solution. It applies the principles of biology and engineering to the development of functional substitutes for damaged tissue. So meaning that in theory, we could take cells from me and that we create an own graft for me. So these grafts are also somehow possible to grow, and they represent a solution for regeneration in the human body, which is not really available. There's no regeneration. Furthermore, these grafts are also anti-thrombogenic. So just to give you like a simple idea of tissue engineering, you see, first step, isolation of autologous cells, cell culture, scaffold, bioreactor, tissue engineered heart valves are ready, implantation. This looks relatively easy. And now I will explain you step by step how this process really works. First of all, we isolate cells. And there are different tissue sources available. For example, we can isolate stem cells from the blood, the bone marrow, or also the adipose tissue, meaning the fat. But also we can use the umbilical cord or amniotic fluid for isolating cells. The cells are then placed in a plastic dish, you can see here, and we feed the cells. So meaning that we provide nutrition so that the cells can grow and proliferate. So we are getting more and more cells. And these cells are then seated on a starter material, or also called scaffold. Importantly here, so the material should be porous so that the cells are able to migrate into the structure. Because the idea is that the biomaterial degrades over time and the cells migrated into the material produce tissue over time. So we have reduced material and an increase of new tissue. In order to stimulate tissue formation, we are placing our material with the seeded cells in a bioreactor in order to simulate the pulsatile blood flow in the human body. You can nicely see how the heart valves are opening and closing, which stimulates the cells to form more tissue. And then, for example, after 6 weeks of culture, you can nicely see the difference between just the plain material, this whitish one, and then the tissue engineered heart valve after 6 weeks, really reddish. So this is new tissue formation. Currently, this tissue engineered heart valves are under preclinical evaluation, meaning that we implant them into an ovine model. And you can nicely see after implantation, so that the leaflets, this is a tissue engineered heart valve, they are opening and closing, and that there's also the blood flow through the heart valve. Besides of tissue engineering heart valves, we are also interested in coronary artery bypass, meaning if some of the vessels are destroyed or closely to blocked, so that we can provide a replacement in order to guarantee the blood flow to the heart. Furthermore, if a myocardial infarction occurred, we are interested in creating a cardiac patch in order to somehow regenerate this ischemic tissue. For this both applications, we are working with the induced pluripotent stem cells. And Dr. Tackenberg already introduced the cells. Just quickly to remind you, Yamanaka discovered them in 2006, and he used just some cells from the skin. We programmed them into induced pluripotent stem cells, and they are such an interesting cell source for us because then you can differentiate them into muscle cells or blood cells. In our case, we are really interested in cells which are important for the cardiovascular system, meaning cells from a vessel, so smooth muscles and endothelial cells, and also from the heart, meaning cardiomyocytes or endothelial cells. These cells represent really a promising tool for the regenerative medicine. And for example, we used some milliliters of blood from a patient, isolated the cells, reprogrammed them into iPSCs, and then differentiated them into smooth muscle cells and endothelial cells, and used them for the tissue engineering application, meaning that we created these tiny vessels. For you, they look quite huge, but they are like 1 centimeter long, and they have an inner diameter less than 1 millimeter. And here, based on the staining, you can see that they have a native-like structure after 6 and 9 weeks of culture. There's quite a lot of tissue formation. Furthermore, as I mentioned, we are also interested in creating a cardiac patch. Therefore, we are using our biodegradable scaffold and seeding induced pluripotent stem cell-derived cardiomyocytes on top. You can nicely see how the cells, our iPSC-derived cardiomyocytes, are beating in a plastic dish. It's a really impressive moment when they start beating. So, and the idea is to create such a patch in order to treat the ischemic damage of the heart. I hope I could give you at least a broad overview of what we are doing. And I also would like to thank all my colleagues which are involved, and also Dr. Tackenberg and Rusland for the collaborative work in our institution, but also all the people and friends already from the SIRA. And I know that also Saito Sensei logged in. Thank you very much. Arigatou gozaimasu.
Thank you very much, Melanie. It is indeed impressive to see the patches moving, and very, very exciting. Thank you very much. I would like to invite the audience again to ask questions. However, my colleague Tomoya Junki has pointed out that maybe Melanie, you can also answer Mr. Yamauchi's question.
Okay.
The original Yamanaka factor was only 4. How many is it currently? And is it free from a cancer generation?
At the moment, so that there are several publications out of it. So they also show that you can remove the CMYC, so meaning this oncogene, for reprogramming. Also, other factors have been used. I think there are even some publications out, they only used 2 factors to produce this iPSCs. In our case, for the blood cells, is a little bit more difficult to reprogram them. And we use the traditional 4 Yamanaka factor and using also the Sendai virus, which is a non-integrative system, which makes it a bit more safe. And yeah, this is a better way in the direction of clinical application.
Thank you very much. Also, thank you, Peter, again for popping up. So in the Q&A, we have hello, Melanie from Peter. It sounds like the tissue engineering already exists, but switching to PSCs, is there a concern about the fetal phenotype? Do the PSCs behave as well as other cells used previously?
So regarding, I hope I got it correctly. So we are doing this differentiation into the cells, and of course, it's always in discussion about the state of the IPC-derived cardiomyocytes, so that they are unmature or like a progenitor kind of state. I must say that we don't see really like a proliferation that we can really keep them on a long term in culture, and they show a mature phenotype. We always also try to compare, for example, for the endothelial cells and smooth muscles, we always compare with a primary cell source.
Thank you. And we have another question. Oh, from Mr. Taki. Maybe I missed it, but how many days does it take to generate these heart valves with, okay, I have to look this up quickly.
Yeah.
Excuse me for the delay. From home cells, how many days?
Yeah, starting with the seeding. So of course, first is expansion time. This really depends on the cells, but starting from the seeding time point until tissue formation, we have, if they are cultured under dynamic, meaning that we simulate the blood flow, because this really improves the tissue formation, at least 4 weeks.
I see. Thank you, Mr. Taki, also for the question. Just a reminder to the audience that you can also send questions in Japanese. Please don't hesitate. We have another question. So how much is the yield in cell number through the tissue process? Is it possible to evaluate?
Not really like exactly. It's more so that we start, we have an average of like 1 million cells per square centimeter, and then they really proliferate and we have this kind of tissue formation. We also analyze later on the DNA amount, but this is really like a linear increase.
I see. Yes, thank you. Would someone also, we have a little bit more time, so would someone also like to ask a question? Okay. In that case, I would like to thank Melanie, Dr. Generali, for her very exciting presentation. And we also look forward to having you at the dialogue session together with our audience. Thank you very much.
Again, Kyoko.
Next, as promised, we will talk from about space. So from Earth to space, also maybe a little bit back down, but I would like to invite Dr. Svante Svante Tauber, excuse me, who will explain to us about learning from space for human health. She's the senior scientist and expert for space life science at the UZH Space Hub, a strategic innovation cluster of the University of Zurich. Dr. Tauber? Can you?
Yes, hello, sorry. Can you hear me now?
Yes, we can.
That's great. I will share my screen.
Thank you.
So thank you very much for the introduction and to speak here to you. I'm very happy about it. So I would like to start by introducing to you the Space Hub of the University of Zurich. So what is this Space Hub? As Christian Sim already mentioned, the University of Zurich is a very innovative university. That means that the strategy is to develop the research into innovations and into products which can be used. The research area of space and aviation has always been strong at our university. And this field of research is by its nature very innovative because the discovery of space is the discovery of the new and also requires new means and strategies. So therefore, in 2018, this research field of space and aviation has been selected as one focus point for innovation at the University of Zurich. So this is called an innovation cluster. And the UZH is the innovation cluster, space and aviation. So what do? We support the research and development in the field of space and aviation at our university. And we establish interdisciplinary collaborations and we connect academia to industry. The goal is to strengthen the space activities in Switzerland by contributing our capabilities and our knowledge. So taken together, we can say that our vision is to use space for innovative research that is useful for society and economy. We're a quite big network at the university. We have more than 25 research groups which belong to the UZH Space Hub, and they are basically from 4 big research areas. There is the area of space life sciences, the big field of Earth observation, there's astrophysics, and there is aviation and drones. So all these researchers, they work on their fascinating projects, which are very diverse. They span from, for instance, space medicine, over biodiversity monitoring, over the discovery or the research on the formation of planets, to the development of autonomously navigating drones. So it's a very wide field, but they all have one in common, and this is a connection to space. So we do not work alone. We have a lot of partners in other research institutions. We also have partners in industry, and we are part of bigger networks inside Switzerland and also worldwide, including also big space agencies such as NASA and ESA. So how do we do experiments in space or in microgravity? How do we do this? One of our research platforms are the so-called Parabolic Flights. This is an established research flight program which is active at the university since 2015, the so-called Swiss Parabolic Flights. So what is this flight? You can see here on this picture a maneuver, which is that an airplane makes a very steep flight into the air, and then it falls down again, coming into horizontal flight. And during this maneuver, there are 20 seconds where everything inside the airplane is weightlessness. You can see this here. You can see the steep flight, looking a bit scary, but it's not actually dangerous. And so, and here you can see the people detaching from the floor when they get weightlessness, and therefore they have to hold on these red straps. So this is the research platform, which is very available for many different research areas, spanning from material science up to space or to gravitational medicine, because researchers can use these 20 seconds of microgravity for their experiments, comparing microgravity or zero gravity to normal gravity. Other platforms that we use at the Space Hub is, for instance, the Zeppelin, which you can see here. This is a big airship which has a very smooth flight and very calm. This is of use for researchers which have very sensitive equipment, for instance. We also have an activity which is the testing of a noise-reduced landing of bigger airplanes, which take place at the UZH Space Hub. And we have big research flight campaigns in the area of global biodiversity monitoring using the so-called Arveris NG spectrometer, which images the Earth and can be used for many different applications. We are also very happy that we have constant access to labs and offices at the Kennedy Space Center in Florida in the USA. Here we have prepared already more than 5 experiments which have been sent to the International Space Station through rockets of the company SpaceX, which operates from the Kennedy Space Center. And for this, we're happy to have collaborations with US entities and companies, enabling this opportunity for us. So I would like now to present 3 research projects more in detail, where we can see that research in microgravity or in space can be beneficial for health on Earth as well. So first, I want to introduce the research of Professor Oliver Ulrich. He's the director of the UZH Space Hub, and he's a professor at several universities, and his focus is the gravitational cell biology. I myself, I'm actually a postdoc in his research group and have had the possibility to participate in experiments during the last 16 years in his group. So the background of the research is that gravitational force has always been present throughout humans' evolution here on Earth. It has shown that human immune system actually is impaired when humans go to space. This has been shown in astronauts. So the question arises in how far Earth's gravity is required for normal cell function and also gene regulation. And how is this very non-specific force of gravity transduced into specific cellular responses? To answer these questions, we have had experiments on several research platforms, for instance, on Parabolic Flights. There we have addressed the question of how cellular signal transduction cascades are affected by microgravity, and we have investigated this with isolated single cells. You can see on the left upper picture some pink fluid. This is actually the medium where the isolated cells are in. These cells are then integrated in bigger hardware racks in the Parabolic Flight airplane, which you can see here. And during the flight, these cells experience microgravity, and then they are chemically fixed, allowing a comparison between cells on the ground under normal gravity to cells in microgravity. Another experimental series we did is the investigation of cellular morphology, metabolism, and gene expression. And for this, we have used a platform which provides 5 minutes of microgravity, and this is so-called sounding rockets. Here you can see actually Professor Ulrich himself working on such a payload of a rocket. And you can also see some cells through the microscope. We also had one very fruitful experiment on the International Space Station. There was the investigation of the macrophage function, and it reveals that actually these macrophages, they adapt to microgravity pretty fast. And this was quite a new finding. So all this research on the gravitational cell biology should in the end help to understand how gravity acts on the human physiology. Additionally, it should help to evaluate the risk and the possibilities for humans to live under different gravity conditions. And they might also help to treat immunodeficiencies or overreactions on Earth. Okay, and the second example of a research project I want to introduce to you is the research of Dr. Jaap Swanenburg. He's a physician at the Biodiversity University Hospital here in Zurich, and he is working on back pain. So back pain is quite a common health problem here on Earth worldwide, and also astronauts face back pain associated with their spaceflight activities. And Dr. Jaap Swanenburg investigates the stabilization mechanisms of the spine under different gravitational conditions to investigate back pain. This is how it works. His measurement equipment is installed in a kind of backpack, which you can see here on the picture. And on this backpack, there is an installation which applies a small push to the back. It's a small one, it doesn't hurt. And in response to this push, the muscle activity is measured, giving information about the response of the back under different gravitational conditions. As you can see here, Dr. Jaap Swanenburg's team, and the aim of his research is to improve the possibilities for treatment for back pain, not only in astronauts, but also here for people on Earth due to a deeper understanding of the mechanisms. The third example I want to talk about is the growing of tissue in space. This is a pilot study which is conducted by Professor Ulrich and his colleague Dr. Cora Thiel from the Space Hub, together with the company Airbus Defence and Space. And in this pilot study, the following strategy is tested. Human adult stem cells are taken from adult humans, and they are brought to the International Space Station where they stay 30 days. During these 30 days in microgravity, these cells form to three-dimensional structures called organoids. These organoids have features of tissue, and the production works better in microgravity than on Earth, where it requires scaffold structures. Several different types of tissue can be produced by this, spanning from kidney up to neurons, and the use of such organoids are broad. They can be used either directly for transplantation and tissue replacement, or they could be used for other in vitro applications, for instance, pharmacological testings, toxicology, disease mechanisms, and they can be used as an alternative for animal models, because these organoids, they are more likely to tissue, they resemble tissue more than just single cells. Yes, with this three examples of experiments and research that are done in space, but they bring benefit to human health on Earth, I would like to end my talk here. And thank you a lot for listening, and I'm open to your questions now.
Excuse me. Thank you very much, Dr. Tauber. such a wide variety of activities and so many meaningful research going on Earth and in space. It's very impressive. I would like to invite the audience to ask questions, or also, since I'm also not an expert, I might not have a question, but maybe your impression about the presentation by Dr. Tauber is also welcome. You can use the chat function or the Q&A function at the bottom of your Zoom screen. Ah, here we go. Wonderful. We have 2 questions. So, okay, thank you. Why do cells form easier 3D structures in space, and how significant is the difference compared to on Earth? No.
Yes. So I think I'm not the best expert on the project, since I'm not working on it myself, but I think the theory why they form better in space is that gravity on Earth always makes the cells sink on the ground, so they are likely to build one cell layer. Whereas in space, you do not have that effect. Cells do not sink on the ground, and therefore they can build 3-dimensional structures more easily because they are not pulled by gravity. Yeah. And the second part of the question, I'm afraid I cannot. That would be a question for the scientists who are really working on the experiment. What is the difference or the similarities to tissues themselves? I can try to reach the researchers and answer these questions in the 30-minute session that we have later. That would be okay.
Yes, I think it was the difference compared to Earth in space.
To Earth.
Yes.
Yeah, then I will try to find out that for you.
Thank you. You mentioned, just going back to the 3D structure, the easiness of forming in space. You mentioned in your presentation that there are scaffolds. So that means you need support more. You need to build additional support?
On Earth, the production of tissues or organoids on Earth needs scaffolds. I think this would be maybe a question for our experts from the ERM. Maybe they can say something about it, because they're experts on growing tissues on Earth.
I see. Okay, so let's circle back to that question at the discussion session then, in that case. But thank you very much for showing us these different activities, so that it inspires our interest. We have a second and third question. Very nice. Well, first of all, thank you, Auno-san, and then thank you, Yamauchi-san, for your question. Microgravity experiments cost much, but huge gravity will be experienced by rotation. Are these knowledges available?
If I understand the question right, this refers to so-called Kleiner rotation. This is a model system on Earth which is applied to model microgravity. And this is a model system or a platform which is available and which is used for experiments about microgravity. But physically, there is a difference between the quality of simulated microgravity by Kleiner rotation and real microgravity, which is achieved by, for instance, Parabolic Flights or the International Space Station. Often in this field of research, first experiments are done by Kleiner rotation because they cost less, this is true, but then they are validated by experiments in real microgravity.
Thank you. Thank you for the answer. Very understandable. And then we have another question from Montoya-san. What other medical applications can best be done in microgravity?
I introduced the three now which are in the scope of our UZA Space Hub. Actually, I can find out to collect some more applications if you want to have additional ones, and then we can circle back to the questions later if that's fine.
Thank you very much for your additional efforts. That would be very nice, maybe in our discussion session then. Yes, so with that then, thank you very much, Svante, for your
welcome
presentation. And we also look forward to discussing with you further in the later session.
Sure.
You. And then I would like to welcome Dr. Claudia Resley. She is the group leader at the Remote Sensing Laboratories of the University of Zurich. She is also the head of the National Point of Contact for Satellite Data, NPOC, and active in science outreach and science communication to foster the use of satellite data for everyone. So very, very fit for our session today. Claudia will inform us about biodiversity from space. What can you see? Dr. Risley?
Thank you, Kyoko, for this introduction. And I'm also going to share my screen. I hope you can see it now.
Yes.
Okay, great. Good. Okay, so thanks also a lot for the invitation and the possibility to speak here and to introduce also the work we are doing. And as you said, so we're coming back more or less to Earth, maybe not physically because we're still staying in space in the method that we're using, satellites, but we are looking back what's going on Earth. I'm part of different groups. So what you heard before is the introduction to the University of Zurich Space Hub. And of course, as I'm working with satellite data, I'm part of this institution as well, of the network. On the other side, I'm the head of the Swiss National Point of Contact for Satellite Images, which is actually an institutional organization to help people dealing with satellite data in Switzerland. So everyone who wants to use satellite data can come to us and ask questions how to get to the data, what to do with it, independent from their background. And with this, I'm embedded in the larger research group of the Remote Sensing Laboratories, which is part of the Department of the Geography within the University of Zurich. So what are we doing in general when we are using satellite data? We are looking at nice images. So what you can see here, you might know it already. So it's the Mount Fuji in Japan. And I'm doing two different parts. So one side, of course, Earth observation, and Earth observation is what we define with observing the entire Earth from space. So we can look wherever we want because the satellites, they look like everywhere. But in particular, it's without touching. So we are not using, for instance, leaves, bring it into the laboratory and destroy them. We are just looking what comes back with the interaction with light, sunlight, or other signals such as radar signals. So what happens if the sunlight touches the Earth and then comes back to the satellite or to any camera or whatever? And also, more important, we are not just looking at the current status of now, but we have the possibility to look back in time. So using different images like from the 80s or from last year or whenever we want. And the title of my project, or the work of my project, is Biodiversity. And also biodiversity, it's quite difficult to define. So there are many different definitions available. And one easy comparison is it's also quite difficult to define climate. So what is climate? Climate is something that is characterized by different things like temperature, surface temperature, water temperature, precipitation, and so on and so forth. And that's the same with biodiversity. So in general, biodiversity is like a variety and variability in nature, maybe. So it's like plants, vegetation, but also animals, insects, whatever, and on different levels. So we're looking at like genetic levels, or not me, but others, which is like the really tiny things. We are looking at species, so individual plants, individual animals, whatever, and then also on an ecosystem level, rather like a large forest as a system, or a country, or a lake, things like this. And we also want to know how they function, so how they work, how they interact, how they benefit from each other. And for sure, what I can say is the negative definition is what you can see down here, like a forest having all the same trees, having all the same heights. That's not what is diverse, biodiversity. So this is the opposite from what we would like to have. In general, I'm working with data coming from the so-called Copernicus program, which is a European program containing, among other data, a lot of space data. So the space component is the central part of this program. And we have this, at the moment, 6 Sentinel satellites, part of the program. My work, I'm mainly working with Sentinel-2, because Sentinel-2 is actually the most, or it's an easy one in order to understand what we see from human eyes and a bit beyond. And that's also the images that I'm going to show you within this presentation. Here, now some theory behind what we are doing. So what you can see on the left side, so we have the wavelengths of the light spectrum. That just means the light that we see contains different parts, and we are looking at the different parts. So for instance, if you're saying what the human eye sees, we are looking up here in these 3 areas, which contain the channels blue, green, and red. That's how our human eye functions, and also for instance, our cell phone functions just with the visible light, we call it. On the other side, we have a very important one area, which is in the near infrared. This is, for instance, where snakes can see. And then on the other side, we have the shortwave infrared, the 2 channels, which are just a bit in another area. And why are we not just looking at this visual area of the light is shown here. This is a so-called spectrum of green vegetation. What do I mean with spectrum? So the plant is using and interacting with the light coming from the sun, and is using the light's energy for growing, so for photosynthesis. And you can see here the signature, meaning, for instance, there is a bump. So there is a bit more reflectance, so the light that comes back to me or the satellite, which has a bump here, which actually corresponds to why we see the plants green. And then here is this low minimum, which is the red color. And then up here, you have a very high level of the near infrared. And when we are looking at the vegetation activity now, we are comparing actually this number here with this number, create a ratio. And if there is a large difference here, so a huge gap, then we can say the plant is using a lot of the red light, the energy of the red light for growing, in comparison with this near infrared. So the vegetation is active. And this is what we do with this so-called normalized difference vegetation index, which is the NDVI. It's just the normalized ratio between the near infrared channel and the red channel. With this representative is coming from Sentinel-2. That's what you see up here. So it's just a satellite that has like band 8 looking at the near infrared and band 4 in the red channel. And this is what I show you now. We're going to use for our work. But first, let's have a look again at Mount Fuji. What can you see from satellite images? So this image is taken on the 5th of August. So it was an overflight from this Sentinel-2. It's the image that we're used to. So it's very green. You see there is a lot of vegetation around the volcano. You see that there is reddish on top and there is non-vegetated area. And then here, a bit disturbing our view, is a cloud here as well. And then you can see also the cloud shadow. If we compare this image now with an image taken from the 24th of October, so a few weeks ago, you can see already it's much darker and it's a bit brownish and it's less green. It makes sense in a way. Of course, now it's autumn towards winter, so vegetation is of course not that active anymore. What you can also see here, there is snow up on the top, and you have again some few clouds covering the peak. But now, to even see it better, as I said before, we are looking at this so-called NDVI, and that is the next image. Here you can see actually not that much, just everything that is dark green means a high NDVI value. Here is the top of the volcano, of course, without vegetation, the clouds as well. You can see some urban area here and the water, which makes sense. But if I compare it now with the 21st of October again, suddenly you see that it's much brighter. So you can start to see areas, I guess, from agricultural areas, and you can see also the valleys. And if I go back and forth again, so there's really a difference in this NDVI value representing the difference of the vegetation activity. Just for completion, I also come back to the image that I showed you before, which is actually using the shortwave infrared as well. So it's another channel in the spectrum. And here you can see, for instance, the urban areas appear suddenly red. And this is also used, for instance, to distinguish between snow appearing in blue and clouds appearing in white, which is a measure of temperature, actually, in this case for the shortwave infrared. So snow is less cold than clouds. Okay, so now we come back to Switzerland, actually, and I show you an example. What does it mean when I talk about this vegetation changes over a season? So what you can see here is a camera looking into the forest, the so-called Leghorn forest here near Zurich. And you can see how the trees become green and then they get colored again red at the end of the season when they lose their leaves. So I play it again, there is some snow. Now the leaf starts to grow. It's getting very green in summer. And then end of the summer, they change color and they lose the leaves again. And winter arrives. And this is the so-called phenology. That's what I'm looking at during my research. So now we change the perspective again. This is again the red, green, blue satellite image of this Leghorn forest, which is actually a very nice west-east face or oriented mountain ridge, which has a steep northern facing slope and a southern facing slope. Which is very interesting for us for monitoring this forest. So if we zoom in into one region of interest, you can see also with the resolution that we are working with. So one of these pixels contains images 10 by 10 meters on the ground. So we can image the entire Earth actually with 10 by 10 meters. As I said before, we are not just looking at one single time, but we are looking at time series. With this Sentinel-2, we are able to get a global coverage every 5 days. So you also get a picture from Japan every 5 days, at least. In Europe, it's a bit more because it's a bit further north, so we have more hour flights due to the orbit of the satellites. How does it look like afterwards? What you see here is what I explained before. So you have this NDVI value in the y-axis, and you have the day of the year on the x-axis. That means each of these either crossed out points, or the points corresponds to 1 hour overflight of the satellite. A high NDVI means high vegetation activity. Low NDVI means low. And what you can see down here, everything that's crossed out is when we detected some clouds, which is of course not interesting for us because we don't see the vegetation. This time series, how we call it, represents now 1 pixel within this picture. And this one represents 1 overflight. And what we do afterwards, we define a mathematical model, in this case a double logistic fit, in order to represent this vegetation cycle over the year. And from this curve that we just fit within these pixels, we extract the start of the season here, what we define as start of the season, mathematically of course, and end of the season here, for instance, or then the amplitude of the vegetation activity. This we do for all the pixels in our area of interest and get then such a map representing the different areas of start of season that we can see. If you do that over a larger image, we can also get a very nice Van Gogh-like paint, which is, again, just representing if you have a bit yellowish areas, that means there is an early start of season, and bluish is a later start of season. What I masked out here, what you can see is this orange and red, black areas, which represents urban areas, of course, no vegetation, and the white ones where we have no pixels, or no valid pixel, means this phenological curve does not represent what we would expect, which can be, for instance, agricultural areas. However, this is not the only type of data that we use from remote sensing, looking at as an input for biodiversity. So this type of start of season we can combine with many other different data sets. And this is a data set coming from my colleague, Isabel Helfenstein, who created a map, also very nice colored, with this, depending on these 3 components here. So if you have rather bluish area, that means these trees are dominated by how much water is available. So this is the leaf water content. Then you have the rather greenish area. That means here chlorophyll is very important. That is a part of the photosynthesis, which means mainly if for instance, the trees are young, so there's young vegetation, they have to grow very fast in order to be competitive. So they have a high vegetation activity in terms of photosynthesis. That's why it's greenish. And then we have also the red one, which is mostly represented at the very top of the ridge, which is carotenoid, which is a safety function of the vegetation towards high radiation. So if there is a lot of sunshine, they protect themselves that they do not get burnt. And then it's getting reddish here. So this is where it's very steep and very south facing. And with all these different data sets and many more, we do then the assessment of biodiversity. So we combine them, for instance, to get an idea of the type of species, so the type of trees, vegetation that we are looking at, about just the vegetation activity, what I explained to you before, then also the health status. So if you look over several years, you can say, okay, now they're less active, so they have issues with, for instance, bark beetles, so animals eating or eating the leaves, what they don't like, of course, the trees. We can also see climate change effects, be it, for instance, the effect of droughts, like we had in 2018-19, very high or a very strong drought in Switzerland. So you can see this effect as well. And that is then again relevant for the further development, for instance, of a forest. As a bit picture, we do the biodiversity monitoring, protection, of course. That's what we would like to have, a good diverse nature, which is also more stable than just a monoculture. Very important for us also to have to make data-driven decisions possible for policymakers. So we give easy to understand information to politicians, and of course monitoring this development goals, the SDG, with our data. With that, I hope I was able to give you a short overview on what we are doing and what you can see and do with satellite data. And with that, I thank you for your attention, and I'm open for your questions.
Thank you very much, Claudia. Very exciting as well. We actually already have a question. Can satellite data generate information on precipitation, ambient temperature, and humidity that will be useful for determining vector density that can predict vector-borne diseases? Thank you, Montoya-san.
Thanks for this question. I'll take the first part. On precipitation, I mean, there are also weather satellites where you see where, for instance, you have rain. What you mainly see with satellite data is the humidity of the surface. So yes. However, if you have clouds, which is usually connected to precipitation, you don't see that much, but you see it afterwards. And you can also switch sensors. So you can switch into the radar domain, which is often used, for instance, for flooding assessments, ambient temperature as well. So there are also other satellites. The picture that I showed at the beginning, so there are 6 different satellites, 6 different types, all of them serve a bit another need. Yes, you can also look at temperature, water temperature, or surface temperature can be done as well. And humidity, what I said before, of course. The vector-borne disease, honestly, don't really know what it means, but what we maybe is related, for instance, if you look, you can see the signature in the data when you see that the vegetation, for instance, is decreasing the activity over the years. You see, okay, there is a disease ongoing, so they're sick, the plants. This can be detected from time series mainly. And as I said, bark beetle, so the little animals that I eat, or they start eating the trees. This can also be detected also with the measure that they become less active, and you see like early signals in order to detect early that they are now infested and invasive species are damaging the trees. I hope that answers the question.
There was an additional note about your question. So it means predict movement of mosquitoes, for example, which carry malaria or dengue fever.
Okay, yes, actually, yes. I just saw a project. It's last week, I think. There is a project ongoing. They developed a prediction of malaria. It's an app that is used in Africa. If you're interested, send me an email. I can find the link again. It's an amazing project. So it's an app that they developed in order to predict the next big outburst of malaria. It's very cool. So just send me an email. I'll find the source.
And you see, can you show your email again?
I think it's still here. No, here.
Excuse me.
So it's just this one. Just send me an email to mpc@geo.uch.ch. I can find it.
Very nice. Very nice. Thank you very much. So thank you again, Dr. Rosalie, Claudia, for your presentation. And I would like to inform the audience that the recording session will end soon. after that, you will have the opportunity to actually turn on your camera. The session will not be recorded. I see maybe, actually, I see another question, but we will maybe take that later in the next session, where the recording will be off. The main point is Science Agora is promoting dialogue between the experts and also our very diverse audience. So you will have the opportunity to ask the questions yourselves after the recording is stopped, with or without the camera on. It is an option, but you will have this chance for the next 30 minutes. But before that, I would like to close this part of the session by thanking everyone, all our collaborators from the University of Zurich for presenting their excellent research from Switzerland. As our title showed at the very end, Science from Switzerland Part 2. We did have another session on October 11th as the pre-Agora event on deepfake. So if you were interested in our presentations today, please also visit the Science Agora website and find the session for October 11. The video is still there and you can watch it on YouTube. It was also a very fascinating and interesting presentation by researchers from University of Zurich and the National Institute of Informatics from Japan. It was a collaborative. Oh, I'm receiving a note. Yes. So once again,